Welcome to another explainer. Honestly, I am absolutely thrilled about today's topic. We're diving into one of the most fascinating, meticulously choreographed processes in all of human biology, the microscopic, clinical journey of fetal development. Just think about it for a second. Every single one of us, you, me, everybody, we all started as just one solitary microscopic cell, right? So how exactly does that single cell orchestrate the flawless construction of a fully functioning human body in a mere 40 weeks? It's basically a masterpiece of biological engineering. And today we're going to walk through the critical milestones of how it all actually happens, step by microscopic step. Let's get right into it.
Okay, so here's our roadmap for today. First up, conception and sex determination. Second, the first trimester milestones. Third, gastrulation, the cellular dance. Fourth, germ layers to major organs. Fifth, the placenta, fetal life support. And finally, number 6, clinical signs of fetal health.
Chapter 1. Conception and sex determination. All right, so this is the starting line. And, the sheer scale and pace of this very first event is it's absolutely mind-blowing. Picture a race, but with roughly 200 to 600 million competitors. That is how many sperm actually begin this fantastic voyage. But out of all those millions, only about 300 to 500 even make it to the ovum in the fallopian tubes. And they can't just show up and be done with it, they have to prepare. First, they go through this thing called capacitation. It's a 7-hour enzyme reaction that strips away their tough outer glycoprotein coatings. Then, right as they reach the ovum, the acrosome reaction kicks in. They release these special hyaluronidase enzymes to literally dissolve the egg's protective outer barrier, the corona radiata. Finally, just a single successful sperm makes contact, fusing with the zona pellucida. And the second that happens, boom, a chemical barrier instantly locks out all the other sperm. The male and female pronuclei fuse and they create a single diploid cell, the zygote.
Now here is a really crucial point. The genetic sex of this brand new individual, it's determined in that exact microscopic fraction of a second. So, the new zygote has 46 chromosomes in total. The maternal parent always contributes an X chromosome, always. So that means the genetic sex is decided entirely by the paternal parent's contribution. If that successful sperm carries an X chromosome, well, it fuses with the maternal X and we get a 46XX genetic female. If it carries a Y chromosome, it fuses to create a 46XY genetic male. It's completely locked in from moment one.
Moving right along to Chapter 2, the first trimester milestones.
The literal moment fertilization happens, the clock starts ticking. We enter this phase of incredibly rapid cellular division as this brand new zygote travels down the fallopian tube toward the uterus. Take a look at the speed of this timeline over just the first week. On day 1, we've got our single-celled zygote, but almost immediately it starts this mitonic division process called cleavage. The cells are rapidly multiplying in number, but interestingly, they're actually decreasing in size. Fast forward to day 3, and we have about 16 cells packed together tightly. It kind of looks like a little mulberry, which is why scientists call it a morula. By day 4, fluid actually rushes into this morula, carving out a hollow cavity and transforming it into what we call a blastocyst. And then, by day 6, this blastocyst has finally reached the uterus and is getting ready to attach.
This brilliantly illustrates that fantastic voyage from the fallopian tubes right down to the uterine wall. But how does the blastocyst actually stick there? Well, it has this amazing outer layer of cells called the trophoblast. These cells are incredible. They essentially act like a microscopic excavation team. They secrete these proteolytic enzymes that actively erode the uterine lining, the endometrium. And this erosion is what allows the blastocyst to embed itself, safely achieving superficial implantation by the end of that 7th day.
All right, Chapter 3, gastrulation, the cellular dance.
Now, this brings us to what is arguably the most critical event of early fetal formation. During the 3rd week, that rapid, frantic division we just talked about slows down a bit. Why? Because the body needs to prioritize delicate, precise structural organization. Welcome to gastrulation. Seriously, if you take away anything from this explainer, let it be this. Gastrulation is like a highly choreographed cellular dance. Or you can think of it like a complex microscopic origami fold. During this crucial 3rd week event, cells on the surface, the epiblast cells, detach and migrate inward along a central groove. They slide underneath to create a brand new middle layer, the intraembryonic mesoderm, sandwiched perfectly between the outer ectoderm and the inner endoderm.
Which perfectly sets up Chapter 4, germ layers to major organs.
So, why does this microscopic origami fold matter so much? Well, because those three newly formed germ layers, the ectoderm, the mesoderm, and the endoderm, they act as the ultimate architectural blueprint. Literally every single tissue and organ in the human body will arise from these three specific layers. We can trace complex human anatomy right back to these exact embryonic coordinates. Let's compare their specific roles. The outermost layer, the ectoderm, essentially forms the central nervous system and the embryonic dorsum, or the back. Then you've got the middle layer, the mesoderm, which builds our skeleton, our musculature, and our urogenital systems. And finally, the innermost layer, the endoderm. That one forms the vital internal linings of major organs, like our respiratory and digestive tracts. It's just absolute staggering precision.
Let's transition to Chapter 5, the placenta, fetal life support.
So while the embryo is busy building a whole body from scratch, it obviously needs absolute protection and constant nourishment. That brings us to the embryonic life support system, the placenta, which is mature and completely functional by the 16th week of development. Let's look at how this builds a bridge between two entirely separate circulatory systems to support fetal growth. In the mature placenta, you have the chorion frondosum on the fetal side, and on the maternal side, the decidua basalis. Remember when implantation happened? Well, maternal capillaries in the lining actually ruptured, and that isn't a mistake, that is completely by design. The maternal blood fills these open areas, which we call intervillous spaces. From there, the fetal blood vessels, running through the umbilical cord, dip down into these pools of maternal blood. This is what allows for the massive, continuous diffusion of nutrients and oxygen, and it does all this without the two blood supplies ever directly mixing.
You see, the placenta is the ultimate biologic multitasker. It establishes this ebb and flow circulation, continuously trading maternal oxygen for fetal carbon dioxide and waste. But it doesn't stop there. It also synthesizes crucial energy sources, like glycogen, cholesterol, and fatty acids, right on site to fuel early fetal growth. Plus, it operates as a full-on endocrine gland, producing maintenance hormones like HCG, estrogen, and the all-important progesterone to keep the pregnancy viable. And incredibly, it actively transports maternal IgG antibodies across the barrier. This means it's actively providing the developing fetus with vital, passive humoral immunity against infections before the baby even takes its very first breath. How amazing is that?
But you know, all of this is happening entirely in the dark, completely hidden from the naked eye. So it really begs the question, from a clinical obstetrician's perspective, how do we actually know this microscopic life support system is functioning properly? How do medical professionals measure the health of this unseen nutrient transfer?
That leads us straight into Chapter 6, Clinical Signs of Fetal Health.
Thanks to modern ultrasound technology and diagnostic testing, obstetricians can actually translate all these hidden cellular functions into highly measurable health markers. When an OB monitors a patient and looks at an ultrasound, they aren't just looking at the baby, they are verifying the entire life support system. They're looking for normal amniotic fluid levels, because an absence of polyhydramnios or oligohydramnios, basically too much or too little fluid, tells them things are functioning well. They closely track steady fetal growth to ensure there's an absence of intrauterine growth restriction, or IUGR, which would signal that the placenta isn't transferring nutrients properly. They also closely inspect the umbilical cord to confirm it's intact with exactly 2 arteries and 1 vein. And finally, through careful testing, they monitor for the absence of viral markers, ensuring that selective placental barrier hasn't been breached by dangerous invaders like CMV, HIV, or toxoplasmosis.
It all comes down to this foundational truth. The source material states it absolutely perfectly. The future health of the individual, in large part, depends on numerous events that take place during gestation. The delicate architectural blueprint laid down in those early, silent weeks of gastrulation, combined with the steady, quiet support of the placenta, they fundamentally dictate our lifelong health and resilience.
Think back to where we started today, from hundreds of millions of sperm down to one single successful fusion. From one solitary cell to a beautifully complex, highly organized, living human being. It really makes you stop and think, if a single cell can build such a flawless, life-sustaining architecture in just a matter of weeks, what other miracles is your body performing right now without you even realizing it? Keep wondering, keep learning, and thank you so much for joining me for this explainer.